My name is Kirti and today we are going to discuss about the topic heredity. Okay, so before going to start our topic heredity, first of all you have to know what is genetics. Do you know what is genetics? Genetics is the branch of biology which deals with the study of heredity, inheritance or variation. Do you know what is variation? Variations means the changes, the differences that you see from your parents to the offspring from one generation to another. That is known as the variation. Let me clear you one thing. What is variation? Variations means changes, or you can say differences in the individual from one generation to another. That is known as variation. Okay, now do you know that variations are of two types? One is the environmental variation second one is the genetic variation. Now, do you know that what is environmental and genetic variations? Environmental variations are those type of variations which occurs due to change in environment. For example, like in our environment, like dust is there, dust, dirt, pollution. So, due to these changes in the environment, the variation which are seen from one generation to another, that variations are known as the environmental variation. Now, what is about the genetic variation? Genetic variations are those variations which occurs due to the change in the germ cells. Or you can say genetic word is derived from the genes, or you can say that genetic variations are those type of variations which are seen due to the change in the genetic composition of any organism. That is known as the genetic variations. Okay, so we are moving to the next point. Next is about what is heredity. Now, do you know that what is heredity or inheritance? Now, first of all, you have to know that what is inheritance or heredity means. Inheritance or heredity means the transfer of characters. You can say the transfer of characters from one generation to another. That is known as inheritance. Means in the inheritance you will see that the characters, or do you know the another name of characters is known as traits. It's known as traits. It means what is heredity? The transfer of characters or transfer of traits that characters which pass from one generation to another, from your parents to your offspring. For example, you will see that you are having some features, some characters similar from your parents, like your height, your skin color, your eyes color, your hair color, that all are due to the inheritance. Okay, now next term is about the genes. Now, do you know that what do you know the term genes? Genes is a segment of DNA that codes for one polynucleotide, that is known as a protein also. So here we can write the segment of DNA that codes for one polynucleotide, or you can say protein. Okay, so it means what is the gene? Genes are the segment of DNA that codes for one protein, or you can say that codes for one polynucleotide. Next, Next is about the chromosomes. Now do you know what is chromosomes? You have seen the chromosomes. Chromosomes are the thread-like structure which are present in the nucleus, and these are present during the cell division. So here we can write it is a thread-like structure which is present in the nucleus of any organism. Okay, that is about the chromosomes. Okay, now next our term is about the phenotype or genotype. I am telling you now one short trick to remember this term. One is about the phenotype, second is about the genotype. So first of all, what is phenotype? Phenotype is derived from a word like pheno. Pheno means like physical characters or physical features. Some are of very tall heighted. So how you will observe this? You have observed about this physical features of that organism. Okay, so that is meant by the term phenotype. Now, but what is genotype? Genotype means type of allele which hides its expression in the presence of dominant allele. Okay, this I will give you example like of pea plant also. Firstly, we are going to tell you about the differences between the dominant and recessive allele. Now, do you know that in the dominant allele, second one is that in the dominant alleles show their effect when an organism is homozygous, dominant or heterozygous dominant. Okay? Means in this case you will tell them that now what is dominant allele? Dominant allele are those type of allele which marks their presence, which hides the presence of recessive allele. And where it will express? Dominant alleles show their effect when an organism is homozygous dominant, or you can say homozygous dominant or heterozygous dominant. Next is about the recessive allele. Now what is recessive allele? Again I am telling you that recessive allele are those type of allele in which they hide their expression in the presence of dominant allele. Okay. Now what is in case of recessive allele? Recessive allele show their effect only in case of like homozygous recessive condition. Okay. Now if I am going to tell you about the example, let's suppose like Mendel has took the example of pea plant. Okay. Now example of dominant allele. Now you can say example like if the progeny means the next generation of pea plant that will be of tall heighted, like capital T pea plant and capital T small t. But if you are considering the example of recessive allele, in recessive allele, you know that small t, and do you know that small t showing for the dwarfness and capital T small t. Let's suppose we are having this type of allele. We are having this type of allele. Allele means like pair of contrasting character. These are a pair of contrasting character now. Like in this capital T showing for the tallness, small t showing for the dwarfness character. So here we can say that this capital T is dominant and this small t is recessive, because in this capital T, this this capital T is a dominant which show their effect and which hides the expression of this dwarfness or this small t. But here you can say that this small t is a recessive. Why it is a recessive? Because these hide the expression of that dominant gene. Okay. Now next I am telling you about the Mendel's experiment. Now do you know that Mendel has conducted his experiment on pea plants? Okay? And on pea plant, do you know what is the scientific name of pea plant? That is known as the Pisum sativum. And during his hybridization experiment, first of all you have to know why it is known as hybridization experiment. Because in this, when they have cross pollination in between two characters, in between these two plants, so they obtain a hybrid character variety. That's why we are known as the hybridization experiment. So first of all, what is Mendel's experiment? Mendel has conducted various hybridization experiment. But when Mendel conducted pea plant, so first of all, a question came in your mind that ma'am, why the Mendel took the pea plant only? Why he not took the other plant? Because pea plant produces large number of seeds in a very small time. Okay, so first point is that why Mendel has conducted pea plant for his experiment, because pea plant produces large number of seeds. Second point, pea plant carry both cell pollination and cross pollination. Okay. And one most important point, that it is easy to transport, that it is very easy to transport also. And next, they show clear-cut differences. Like they show clear-cut differences, that character they have studied. So first of all, why Mendel has conducted experiment on the pea plant? Because, and one more point is there, easily observable characters. Easily observable character. Okay, so these are the conditions that Mendel have to require for his pea plant experiment. Okay. Now, one more term is there. One is homozygous and heterozygous. Now, do you know that homozygous? Homo means like similar. Homo means homozygous is derived from a word like homo. Homo means same. And hetero means different. Hetero means different. So first of all, what is homozygous and heterozygous? Homozygous is that condition which are having same type of genes. But what does go about in the heterozygous? Heterozygous means which are having different types of genes. Okay, which are present in alleles. Which alleles are the pair of contrasting character. Okay, that is known as the homozygous and heterozygous. Let me explain you by example. Let's suppose if someone is giving you one example, like we have to identify that this is a homozygous or heterozygous gene. So, like I am writing capital T. Okay, so in this you can easily observe that ma'am, these are two same type of genes. So this is a homozygous condition. If I am writing small t, small t. So you can easily say that ma'am, these are also two, they are also having the same type of gene. So they are, it is also a homozygous condition. Okay. But if I am saying that capital T, small t, now in this case, they are having both type of genes. They are having dominant gene also. They are having recessive gene also. So here we can say these are heterozygous condition. Okay, here we can say these are heterozygous. Hetero means they having two types of genes because these are dominant gene. This one is a recessive gene. Okay, so this is about the homozygous and heterozygous condition. Now our next topic is law of dominance, which is under the Mendel's law of inheritance. Now, do you know that what do you mean by the term law of dominance? As to explain its inheritance concept, to explain its heredity concept, Mendel has given some laws. First law is about the law of dominance. So first of all, what do you mean by the term law of dominance? Like law of dominance states that during F1 generation, means first of all, what is law of dominance? Law of dominance is क्या law of dominance states that during F1 generation, means when the two pea plants are cross pollination among them. So when they are producing F1 generation, the alleles of both plants, or you can say the alleles of both parents showing their own effect. That is known as the law of dominance. Let me give you one example for this. Inheritance of study for one contrasting character. For one contrasting characters. Now, do you know that what is one contrasting character? One contrasting character means that Mendel has selected only one character. For example, and do you know one contrasting character study is also known as the monohybrid cross. That is also known as monohybrid cross. Okay. Now I am going to tell you about that study of inheritance for one contrasting character, or you can say for that what is monohybrid cross. I will analyze it. Monohybrid cross. So beta, first of all you have to know that what is monohybrid cross. So first of all, Mendel has selected one character of pea plant. That Mendel has selected character, one contrasting character that selected plant height. Plant height for monohybrid cross. Okay, first of all, he took pea plant height like one he considered tall heighted. Other he considered of dwarf heighted. Okay, and he took experiment that is cross pollination. Cross pollination is that among the two different. Okay, so for the tall heighted, we can write like capital T. Okay, because these both are the homozygous. You can write it homozygous. And for dwarf variety, what he took? Small t, and it is also a homozygous condition. Okay, homozygous condition means, or homozygous alleles means which are having two similar type of genes. Okay, now when they are producing gametes, means during the process of division, during the cell division, these producing gametes. We can write small t, here we can write Here we can write capital T and here we can write small t as well. Okay. So these are the gametes. Now, when they done the cross pollination like this way, when they done the cross pollination, you will observe that F1 generation. Now, first of all, you tell me that do you know that what is F1 generation? F1 generation is the cross in between these two organisms. In between F1 generation, you can say it is the result of cross pollination. Okay. So here we know that we observe that F1 generation will be capital T small t, and this capital T small t denotes the tallness. This is denoting the tallness. And do you know that here this capital T is dominant? Dominant. Why it is dominant? Because it's showing the tallest character. It's showing the tall character. Here the capital T is dominant, and it shows their effect in the presence of this recessive allele. And this recessive allele hides its effect in the presence of dominant allele. Okay. Now, this one is the result of F1 generation. Now we have to produce the F2 generation also. So do you know that how we obtain the F2 generation? We obtain F2 generation by the self-pollination of these F1 generation offspring. Means when we are getting the self-pollination among two same F1 generation offspring, we are getting the F2 generation. And F2 generation is the result of self-pollination. Okay. Now I am telling about the F2 generation, how we will obtain the F2 generation. Next is self-pollination among F1 generation offspring. You can write, or you can write progeny also. Means offspring or progeny tell you about the next generation. Okay. Now, self-pollination among F1 generation. Why we are running the self-pollination? Just to produce the F2 generation. Here we can write like the parents. Because now they are including the parents like capital T, small t. Here we can write capital T, small t. Now, do you know when these are forming the gametes? When they are forming gametes, it again split capital T, small t, capital T, small t. Okay. Now, these are the gametes. Now you have to produce the F1 generation offspring or progeny. F2 generation offspring or progeny. So first of all, you have to take this capital T of first parent fuse with this capital T of second parent. So what product it will form? Capital T, capital T. Next, this capital T again fuse with this small t, it will form capital T, small t. Next, this small t of first parent fuses with this capital T, it again forms capital T, small t. Next, this small t fused with the small t, it again forms the small t, small t. Now we have to determine the ratio, like phenotypic ratio and genotypic ratio as well. First of all, you have to know that what is it? It's showing the tallness or dwarfness. It's showing for the tallness. In the same way, these are also showing the tallness. These are showing for the dwarfness or dwarf hybrid plant. Why it's showing for the tallness and dwarfness? Because here you will see that this capital T is dominant. Because these are same type of gene, and same type of genes are known as the homozygous. This showing the homozygous condition. In this, you will see these both are having two different types of genes. If they are having two different types of genes, it means they showing the heterozygous condition. They showing the heterozygous condition. Now, here you will see this showing for the dwarfness. If they are showing for the dwarfness, it means they are showing the homozygous condition. Why showing for the homozygous condition? Because they are having two same type of genes. That's why they are showing for the homozygous condition, and these both showing for the heterozygous condition. Okay. Now next we have to calculate about the phenotypic ratio and genotypic ratio. Now, do you know that what is phenotypic ratio and what is genotypic ratio? As we have already discussed, that phenotype means the physical characters of any organism, that is known as the phenotypic characters. But what is genotypic characters? Genotypic characters means the genetic composition. So on the basis of that characters, we will identify what is phenotype characters and what is genotype characters. So first of all, on the basis of phenotype. Now in this cross, in this monohybrid cross, in this inheritance of study for one contrasting character, we have considered one character, and on the basis of that we see that it's showing for tallness versus dwarf height, or tall heighted versus dwarf heighted. Okay, so here you will check tall heighted. How many offsprings are there, or you can say how many progeny were from tall heighted. Here you will see like this, capital T, because they are showing for the tallness. This also showing for the tallness. Why they are showing for the tallness? Because here capital T is dominant over that small t. Here also capital T is dominant over that small t, because here capital T is dominant because it's showing their effect. It's showing for the tallest character. So here 1, 2 and 3. 3 are tall heighted and remaining one is of dwarf heighted, because these are shown for the dwarfest character. So here our phenotypic ratio will be 3:1. But now we have to identify the genotypic ratio. Genotypic means the genetic composition, the genetic level. So how we can identify the genotypic composition or genotypic ratio on the basis of these generations? Like we can say capital T versus capital T versus small t. Okay. Now we have to identify how many offsprings are from capital T, 1. These are 2 and these last are 1. So here the genotypic ratio will be 1:2:1. Next. So I am again telling you, phenotypic ratio will be, as it was mess up. So I am again writing here. Phenotypic and genotypic ratio. Phenotypic ratio means like physical features. So here we can write tall versus dwarf. Now you will tell me what is the ratio, phenotypic ratio? That is 3:1. Now what is genotypic ratio? In that monohybrid case, it is capital T versus capital T versus small t. Okay, these are homozygous condition, these are heterozygous condition. Again, these are showing the homozygous condition. Now, for that case, genotypic ratio will be 1:2:1. Now, in that case, our monohybrid clause is finished. Okay, now, beta, our next topic is study of inheritance for two contrasting characters. Now with a study of inheritance for two contrasting characters, it means that Mendel has selected two contrasting characters for his experiment, for his study, and that study of two contrasting characters is also known as the dihybrid cross. That is also known as dihybrid cross. Why it is known as dihybrid cross? Because they have considered two characters at one time. Let's suppose like in monohybrid cross, they have took only one character, that is of plant height. But in that case, they consider two characters at one time, like they are considered two characters: seed color and seed shape. Seed color and seed shape. So first of all, beta, they have considered two characters at one time. That's why known as the study of inheritance for two contrasting characters. Now, what seed color and seed shape they have took for this experiment? First of all, you have to know that brown, wrinkled, yellow and green. Now you have to know that round seeds denote with the capital R. Okay, so first of all, before conducting this experiment, you have to know that round seeds denoting for capital R and wrinkled seeds denoting for the small r. Yellow color of that seed denoting for the capital Y and green color of that seed denoting for the small y. Okay, now we are starting from the dihybrid cross. We are starting here like for dihybrid cross. Firstly, the selected parents having two contrasting characters like brown yellow and brown yellow and wrinkled green color seeds. Okay, so as I have already told you that for round seed shape denoting for the capital R, here we can write, and for yellow color seeds denoting for the capital Y, and for wrinkled green it's showing for the small r, and for green color seeds it's showing for the small y. It means round and wrinkled denotes for the seed shape. Yellow and green denotes for the seed color. Okay. Now next is when they are producing the gametes. It will be capital R, capital Y. It will be small r, small y. Now beta you can say that now can we take round green and yellow and round green and wrinkled yellow. Yes, you can take this example also. Like somebody is taking like round green color seeds on one side, somebody is taking like wrinkled yellow seeds on other side also. So it's your choice, you can take this also. So first of all beta, next in between these two gametes, what we have to do? We have to do the cross pollination to produce the F1 generation. As I have already told you that F1 generation is the result of cross pollination. Okay, so when we are doing the cross pollination among these two gametes, what it will form? Capital R. Firstly we have to write the capital, then small r, then again capital Y, and then again small y. So here you will tell me, can you tell me that this F1 generation representing which type of seed, which type of color? So first of all you will see that this capital R, it is dominant over that small r. This capital Y is dominant over that small y. So here F1 generation will be brown, yellow. Okay, brown, yellow color seed they are showing. Okay, so next one is when we have to produce the F2 generation. So do you know that how we can produce the F2 generation in between these dihybrid cross? So this one is the result of F1 generation of dihybrid cross. Now we are moving for the self pollination. Now we have to do self pollination in between two F1 generation offspring, because these are the results of F1 generation offspring, and in between the self pollination, in between these two self pollination, we have to obtain the F2 generation offspring. So how we can obtain the F2 generation offspring? First of all beta, when they are producing the gametes, because due to the self pollination they are producing gametes also. So how many types of gametes they will form? I will explain you by making a table also. Like capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These four types of gametes which are formed by the self-pollination. Here we can also write here like in this way: capital R, capital Y, capital R, small r, capital Y, and small r, small y. Okay. And sometimes this cross is also asked in your board exam also in 10th class. So first of all, you have to do self-pollination among these gametes like this one. This capital R fused with this capital R, capital Y. We can write it like that. Next, capital R, and first you have to write the capital word, then you have to write the small words. Capital Y, small y. Then again capital R, capital Y, small y. Then again capital R, capital Y, small y. Okay. In this way we will do all this: capital R, capital Y, small y. Again capital R, small y. Next, capital R, capital Y, small y. In this way, listen. Same way, capital R, small r, capital Y, capital R, small r. What is it? Why it's closed? Wait. It's still moving. Wait for 1 minute. So we continue like with the fusion of these two gametes, small r, again capital Y, small y, and last this capital R, capital Y, small y. This capital R, small y, small y. Again this small r, capital Y, small y, small r, small y, small. Now beta, you have to tell the ratio in between these four gametes. So how you will calculate the ratio? Let's suppose you are writing here like small r, capital Y, ratio small r, small y, ratio small r, small y, ratio small r, small y. So here, be careful. First of all, how you will identify the small capital R, capital Y in this? First, we check it. This one is off because capital R is dominant, capital Y is also dominant here. So first is this, we will count it. How many capital R and capital Y is there in our ratio? 1, 2, 3, 4, 5, 6, 7, 8 and 9. Okay, so here we can write 9. Now we have to calculate capital R and small y. So here we will check capital R and small y. 1, 2 and 3. So here we can write ratio 3 of capital R, small y we can write 3. Next one is small r and capital Y. You can check here small r and capital Y. 1, 2 and again 3. Here we can write 3. Now in the last you have to check small r and small y. Now only one is there. That's showing the small r and small y character. So its ratio will be 9:3:3:1. Okay. Now our last topic is about the sex determination. First of all, what is sex determination? Sex determination is the process of determining the sex of any organism, means either develops into male or they develop into female child or an organism. Okay. So first of all, beta, you have to know that what is sex determination. Sex determination is the process of determining the sex of any organism, that it develops into a male child or it develops into a female child. Okay. So first of all, we are considering the case of reptiles. Now then after that we are moving in case of human beings also. But firstly, you have to know in case of reptiles. Now, do you know that in case of reptiles, sex determination is influenced by some environmental factors? Do you know that in case of reptiles, the sex determination is influenced by the environmental factors also? Okay, like if we are considering then the case of temperature, because temperature is also an environmental factor. So if we are considering the case of temperature, for example, in turtles, we are considering example of turtles and lizards. In case of turtles, if they are occupying their eggs at a very high temperature, so due to the very high temperature, mostly turtles will develop into the male. If they are incubating their eggs at a very high temperature. But in case of lizards, if they are incubating their eggs at a very high temperature, so most of the lizards will convert into the females. So here you can see that temperature is that environmental factor which determines the sex of that reptile in case of turtles and lizards. Next, we have to discuss in case of human beings. In case of human beings, so first of all, in case of human beings, do you know that in a human being, how many number of chromosomes are present? There are total 46 number of chromosomes are present in case of human beings, in which it is present in the form of 23 pair. 23 pair is present there. So first of all, in human beings, do you know that in males and in females, in males there are two types of chromosomes are present. One is the X chromosome, second is the Y chromosome. And in case of females, they are having two same type of chromosome. It means two X chromosomes are present in case of females. Okay. So if the males are having two different type of chromosomes, so you can say that this is a heterogametic or heterogamete. But hetero means it means two different types of chromosome or two different type of gametes. But if they are having two same type of gametes, you can say it is homogamete or homogametic. Okay. So we have to discuss about the sex determination in human beings. That in human beings, how it is determined that the next generation or next progeny or next offspring will develop or develops into the male or in case of female. So now I am going to tell you that like male and female, we all know that like in case of males and female also they are having 46 number of chromosomes. So here we can write like in this way, 44 plus XY. In case of female, we can write 44 plus XX. Why we have written this like that? Because total number of chromosomes are 46 in each. 44, and why we have written it XY? Because males are having two different type of chromosomes, that is X chromosome and Y chromosome. But in case of females, they are having two same type of chromosomes, that is X chromosomes. Okay, same type of chromosomes. Now, when they are forming the gametes, when they are forming the gamete production, so but it will form 22 plus X. Again 22 plus Y, these are the gametes. But when the female producing the gametes, it again forms 22 plus X and 22 plus X again, because these are the homogametes and these are the heterogametes, because they are having two different type of gametes. They are having two same type of gametes. So here we can write this one is the heterogametes and this is the homogametes. have to produce the next generation, they have to produce the progeny. So how they will use? Like this 22 plus X of male, means this gamete of male fuses with the female gamete. So it will form 44 plus XX. Okay. When this again, this fuses with this gamete, it again forms 44 plus XY. We can write like this also. Now again this gamete fuses with this, it again forms 44 plus X. Now this gamete of male fuses with this female gamete, it again forms 44 plus XY. So here you will find that which how many possibilities of male and how many possibilities of female child. So here you will see that 2X means XX, that is having a homogamete. These are having two same type of chromosomes, so it will develop into a female. Next, XY, two different chromosomes. X and Y both are different chromosomes, so it will develop into a male. Here again two X chromosome, so here it develops into a female. Again, last X. This is the last topic. XY, because these are having two different chromosomes, so it again develops into the male. So here you will check that how many possibilities of female child and how many possibilities of male child will be there. Like one female, it means two females. One male and one male, it means both are having 50% chances of male child and 50% chances will be of female child. Okay. So if you are having any doubt, you can comment in the comment section box also. And with this, our chapter is going to finish also. So thank you. Have a nice day.